Hey guys, in this video we're going to talk about diffusion and osmosis. Both of those deal with the movement of materials. We're going to start with diffusion. And what I've got here is a picture of a cell. And if you know anything about cells, which you should know that this is a eukaryotic cell because it has a nucleus. And you should know that it is an animal cell because it lacks a cell wall. Two key characteristics to tie into some other things. But in this animal cell, we're going to look at a couple key things. First off, the cytoplasm. So right over here I've got it highlighted for you. And the cytoplasm is what I like to call the chicken noodle soup of the cell. It's got everything you need, but it's all just kind of mixed in there into this fluid called the cytosol. So if the cytoplasm is chicken noodle soup, which has all kinds of stuff mixed into it, the cytosol is the broth. And this little broth mixture, everything is mixed in that the cell needs. But I hope you can see the problem that might create. You might have a whole bunch of chicken over here, but people really would like some chicken over here. So how does the chicken go from here to here in the sense of the chicken noodle soup? Well, that problem doesn't actually happen in a cell because there's no chicken inside of a cell and there's not really a soup. But in that cytoplasm, there are things that are needed. And how do they get from one location to another is a very important thing called diffusion, which is the movement of materials, of solute, from areas of high concentration to areas of low concentration. Sadly, one of the best examples of this is, well, quite frankly, a fart. If you think about when someone passes gas, in the area that they release it initially, it's in very high concentration and it smells bad. But as time passes along, that smell then dissipates. It's not that it goes away, but simply it spreads to the areas of low concentration. Sadly, although it's a crude example, it is a good example of diffusion in an air sense. A very similar thing happens inside a cell, to where certain things that might be located in this region, if it's high concentration, will move to areas of low concentration. And so very basic, that's diffusion. And it is a driving force behind the movement of many substances across cell membranes as well. And so what I mean by that is that some things that will be inside of the cell will actually move out of the cell because of this diffusion pressure. So let's suppose we're looking at unequal concentrations of something across the cell membrane. And if you remember, this cell membrane right here has a big hydrophobic layer, so that things don't like to move through it. But if we take a look, we've got much less solute on this side of the membrane than we have on the other side. And so what will happen over time is that there will be this diffusion pressure, where this is high concentration, and this is low concentration, and things will move from high to low, and so we'll have a net movement in this direction. If we continue and look at that picture, we'll notice that we have some arrows moving in our net direction. I'll draw those in red. That's 3 moving in our net direction, but we also, in blue, have 1 moving against our net direction. And so if we look, we do have particles moving in and out of this membrane because they can pass through it. Most of them are moving in, which gives us net movement into the cell. And this is what diffusion does. It moves things from high concentration to low concentration. If we continue, if we let it go longer, what we see is that they eventually reach this thing called equilibrium, where we have equal numbers on either side. So if we look, we have 10 here and 10 here, which means there's no high, there's no low, so there's no net movement. I say no net movement. You'll notice that in this particular picture, we have 3 arrows. Rather, 4 arrows. 2 moving in, 2 moving out. And what that represents is something called dynamic equilibrium, where substances move in and out. But as one substance moves into the cell, another will then move out because it's going to keep that equilibrium. If cells reach dynamic equilibrium, typically they're dead. And so your cells are going to work very hard at controlling diffusion and using it to their advantage. But it basically runs off this very simple premise of things will move from high concentrations to low concentration until equilibrium is reached. This type of transport, where equilibrium is reached, is diffusion, but it's also what we call passive transport. And it is passive because it uses no energy. So again, no energy required for this, it just happens on its own. They're going to move in both directions. Just because you've hit equilibrium does not mean nothing's moving. If we talk about facilitated diffusion, it's a type of passive diffusion, which means since it's passive, it does not require any cellular energy. But it's actually going to use some protein assistance. So we really see 2 main types. We see the one over here, And we see the example here with like a protein channel. These are two examples. And so what we have is we have this protein, and this protein is going to allow things to move in and out. You'll notice that this particular substrate will move in. It doesn't require any energy. It's just simply going to, allow that one substrate to move. It kind of changes shape here, and then, it spits out the other side. Shape is very important to membrane function, and so it's basically judging this particular particle and rejecting that one because it's very specific in that case. Our channel protein is a lot less specific. We have an area, and if you can fit in that area, you can move right on in. So you'll notice that in this case, the small particles are allowed to go in, but the larger particles which cannot fit are not able to go through there. Facilitated diffusion, like I said, does not require energy. It does, however, require these proteins. If something were to go wrong with this channel protein, it would not be able to move things across the membrane, because if you remember, this cellular membrane has this hydrophobic layer where nothing will want to move through it. And so because it has that hydrophobic layer, particles need these proteins to move through. Facilitated diffusion, in essence, gives you a bridge across this hydrophobic layer, kind of like a bridge crossing a waterway. If the waterway's too dangerous, you can't swim across it. But if you put a bridge there, you can easily now walk across that bridge. That's the role of these proteins in facilitated diffusion. Osmosis is a very specific movement of water, and it's going to use facilitated diffusion because in living organisms, water can't move across this membrane, so we're going to use these aquaporins. And aquaporins allow water to pass right through the membrane and through this water-hating or hydrophobic layer. And so, again, this is just a channel protein. So you notice it's got a size, and water can fit through that size. So water will shoot right through, and it will do so moving from high concentrations of water to low concentrations of water. Now, we're going to watch this video. And this video is taking a raw egg that has had its shell dissolved off of it. So if you take a look here, this is the egg, and we've dissolved the shell off of it, so it's no longer on there. And what we've done is we've stuck it into a sugary solution. So basically we're going to cause water to move, and since it's a sugary solution, what ends up happening is this is very low water, and in here is high water. And so if you know osmosis, you'll know things move from high water concentrations to low water concentration, so they're going to end up moving out. So we're going to see this egg shrivel up, and then we're going to flip it. We're going to put it into water, which is opposite. We're going to see it expand. So osmosis is very important, and typically it's moving across a selectively permeable membrane. Selectively permeable means that it's selecting what things can move through. Permeability, or permeable, is a word that means able to move through. Doorways are permeable parts of houses. So selectively permeable membranes allow some things to move through where they don't allow others. So let's take a look at this video. It is a time elapse. We're looking at 15 minutes. Progress to 30. Now you can really see it shriveling up. Now we end up with 45. And at this hour mark, we can really see it super shriveled up. And what they're going to do is they're going to flip it to distilled water. And so at this point, you can really see that a lot of that water has moved out. Again, flip it to distilled water, and we watch it expand. Again, it's refilling up with water. That selectively permeable membrane allows water to move through. 15 minutes, and now you're going to see the size increase, which is going to swell over time. This eventually is going to be allowed to go for 2 hours. And if you really pay attention, you can see it growing and growing and growing. And that's as more and more water are filling into this egg. Again, at the very first part, we took water out by putting it in a sugary solution. At the end of it, now we've got it in distilled water, which means that the low water concentration is actually inside the cell, and high water concentrations out here in the water. And so water's going to rush in and rush in. Again, this video only goes for about 2 hours of time elapse, but what it shows you is the movement of water right through a true living membrane inside this raw egg. It's pretty neat because, and again, you can see it in time elapse happening. Now, like I said before, if we talk about concentrations, with respect to water, out here is now high because we have a distilled water, which is pure water. And it will move into here because in here is low. It really depends on what you're sticking a cell or an organism into as to what kind of osmotic pressure it's going to hit. We're going to talk about 3 main types of situations. Osmosis really is quite simple. We're going to have to compare solute to solvent and where water is compared to what's mixed into water. This particular experiment is quite classic. Red indicates your solute or what's mixed in there, And so you have very low solute over here and very high solute over here. So your solute should want to move in this direction. But we've got this semi-permeable membrane that doesn't allow that to happen. So since the solute can't move, our water will. Ironically, water is opposite. So if solute concentration is low, water concentration will be high. And if solute concentration is high, water concentration will be low, which means water wants to move this way, and because our selectively permeable membrane allows water to move, it does so. So if we progress through time, very sloppily written, what we find is that the water level over here has dropped. And that's because water has moved across this membrane. This is actually the exact same thing we saw in our egg. The actual solute that was inside that egg couldn't move, so the water moved. And that caused the egg to shrivel up. On this side right here, it's like the simulation of the shriveling up egg. It has so little solute in it compared to what's outside of that particular cell that it's going to move out of there. In general, that's how osmosis works. And if you look over here, we're taking a look at the permeability of this membrane. The aquaporins allow water to move, but these bigger sugar molecules or other macromolecules inside of a cell, they can't move. So only water is able to move. That's why we saw that egg shrivel and swell. Osmotic pressure is the force that actually makes this happen. And we're going to look at a couple situations. First off is what we call isotonic. And basically what isotonic is wherever a cell is in, a good concentration inside and outside. It's equal. And so what we'll find in isotonic is that water will move in and out of a cell. Cells are perfectly happy in an isotonic solution. So here we have a red blood cell, and here we have a plant cell. And this red blood cell can be placed in plasma. It's very happy. Water's going to move in and out, no problem. If we place it into a hypertonic solution, and the prefix hyper should make you think high. A hypertonic solution has a higher solute concentration. So what that means is that since out here is high for solute, inside will be low for solute. And if we identify water, that means out here is low and in here is high. And so because hypertonic solutions have different solutes, what we'll see is that water will move outside of the cell. If you remember nothing about hypertonic, remember that water molecules move out of the cell in a hypertonic solution. And this is where we took that egg and we stuck it into the sugary solution. That was a hypertonic solution, and we watched the egg shrivel up. That's because water moved out of it due to the fact we put it in a hypertonic solution. We then flipped the experiment and we put it into a hypotonic solution. And what that means, hypo should make you think low. And so what happens is you have a lower solute concentration outside the cell. So if we identify solute, out here is low and here is high. Again, opposite for water. So if outside's low, water concentration will be high out here, and it must be low inside the cell. And what that will cause is water to move from the outside of the cell into the inside. Again, if we think about our video, this is when we took the shriveled egg and we put it into that distilled water. Distilled water has very, very low solute concentration because it's just plain old water. Since the solutes can't move out of the cell, water is going to go rushing in. And that's where we saw the cell expand and then eventually just get bigger and bigger and bigger. If you take red blood cells like the one we see here and you place them into a hypertonic solution, they'll shrivel up because water will move out. If you then take them out of that hypertonic solution and put them into this hypotonic solution, just like we did the egg, that shriveled red blood cell will swell back up and it'll eventually burst. So many cells, unless they have a cell wall like this plant cell right here, they will burst in a hypotonic solution. Again, if you remember nothing of these things, remember that a hypertonic solution, water moves out. Hypotonic solution, water will move in.